All right, let's jump right in. Today, in this explainer, we are exploring the absolutely fascinating biochemistry of carbohydrates. We're going to see exactly how simple sugar building blocks power your muscles during a heavy workout, and, incredibly, how those exact same types of building blocks are used to construct towering oak trees. It's just a brilliant story of molecular design, so let's get started.
Here's a quick look at a roadmap. We'll start by meeting the carbohydrate family, then look at making and breaking maltose. After that, we'll dive into starch for plant energy, glycogen for animal energy, and finally, wrap up with cellulose as nature's structural support.
Okay, part 1. Let's meet the carbohydrate family.
So the crucial thing to understand here is that all carbohydrates, whether we're talking about a teaspoon of table sugar or the trunk of a massive tree, they all follow a very strict chemical blueprint. They are composed entirely of carbon, hydrogen, and oxygen atoms, and they always appear in a very specific, fixed ratio of 1 to 2 to 1. This gives us that universal molecular formula of CH2ON, where N simply equals the number of carbon atoms. So, if N happens to be 6, you get C6H12O6, which you probably recognize as glucose.
And this perfectly illustrates our three main classes based on that blueprint. On the far left, monosaccharides, things like glucose, fructose, and galactose. Think of these as your single, simple sugar units, basically your individual Lego bricks. In the middle, we have disaccharides like maltose, which are exactly 2 of those bricks snapped together. And then on the right, we have polysaccharides like starch, glycogen, and cellulose. These are massive, complex polymers made up of just tons and tons of monosaccharide bricks joined together.
Let's move to part 2, making and breaking maltose. So how do we actually snap these individual bricks together? Well, it happens through a process called a condensation reaction. Here's how it plays out. First, 2 individual alpha-glucose monomers align right next to each other. Then, a chemical reaction occurs that specifically kicks out one molecule of water, H2O. And finally, the removal of that water allows the 2 glucose molecules to bond together. This forms what we call an alpha-1,4 glycosidic linkage. And boom, the result is our disaccharide, maltose.
You can literally see that chemical snap happening right here. This perfectly visualizes how removing the OH from one glucose and the H from the other creates that water byproduct. The oxygen atom that's left over acts as a bridge, forming that alpha-1,4 glycosidic linkage. And just as a quick reminder, we call it 1,4 because it bonds carbon number 1 of the first glucose directly to carbon number 4 of the second one.
But biological systems are highly dynamic, right? If your body suddenly needs a quick burst of energy, it has to break that maltose back down into usable single glucose units. And we do this by just throwing the entire process in reverse. We use a reaction called hydrolysis. By introducing a water molecule back into the mix, that alpha-1,4 glycosidic linkage gets severed, separating the dimer back into 2 individual alpha-glucose monomers that are ready to be burned for fuel. This is hydrolysis in action. Notice how the process is pointing the other way now. The word hydrolysis literally translates to water breaking. We add the H2O back across that oxygen bridge, the bond breaks, and we return exactly to our 2 basic building blocks. Honestly, this reversible snapping and unsnapping is the fundamental mechanic that drives carbohydrate biology.
All right, section 3, starch, plant energy storage.
Starch is the main carbohydrate storage in plants, usually tucked away in organelles like plastids and amyloplasts. Now, what makes it the absolute perfect plant battery? Well, despite being physically huge, it's a very compact molecule. But even more importantly, it's insoluble in water. This is a massive, game-changing advantage because it means storing all this energy doesn't mess with the plant cell's water potential or alter its osmotic pressure. And when the plant actually needs energy, those glycosidic bonds can be easily hydrolyzed. Now, starch actually comes in 2 totally distinct architectural forms. Over on the left, we have amylose. Amylose is an unbranched polymer. It consists solely of alpha-glucose monomers joined by alpha-1,4 linkages, and this specific structure causes it to coil up into a helix. Then on the right, we have amylopectin. Amylopectin starts with that exact same alpha-1,4 chain, but periodically it uses an alpha-1,6 glycosidic linkage. And that 1,6 bond creates a branch point, giving amylopectin this really complex tree-like structure.
Moving right along to section 4, glycogen, which is animal energy storage.
So what about us? Animals rely on a polysaccharide called glycogen.
Glycogen serves as our main carbohydrate storage, stockpiled mostly in your liver cells and your muscle cells. Just like starch, it's a polymer made entirely of alpha-glucose monomers. But structurally, it is evolved in a very specific way to meet the high, rapid-fire energy demands of constantly moving, breathing animals.
Let's see how this builds on what we just learned about plants. Glycogen uses the exact same chemistry as a plant's amylopectin. It has linear chains of alpha-1 to 4 linkages and branch points made of alpha-1 to 6 linkages. However, glycogen is significantly more branched. Why does this matter? Well, because enzymes break down these polymers starting from the ends of the branches. More branches mean more available ends. This highly branched architecture allows an animal's body to snip off thousands of glucose bricks simultaneously, giving us that rapid, on-demand energy in a fraction of a second.
Finally, Section 5, Cellulose, Nature's Structural Support. This is where a tiny little monomer tweak changes absolutely everything. Instead of using alpha-glucose, cellulose is made entirely of beta-glucose monomers. And because of the specific shape of beta-glucose, when they join together via beta-1,4 glycosidic linkages, they don't coil up, they don't branch. Instead, they form a long, rigidly straight, unbranched chain. Literally every alternating glucose molecule is essentially flipped upside down to make this straight line physically possible.
And what's really fascinating is what happens when you put multiple straight cellulose chains right next to each other. Because they are so perfectly straight, they line up parallel. The hydroxyl groups protruding from the chains lock together with surrounding chains using hydrogen bonds. This forms incredibly strong rope-like bundles that we call microfibrils. These microfibrils are woven together to provide the major structural support in plant cell walls. It's quite literally the reason a tree can stand hundreds of feet tall, and it's what gives a stalk of celery its crunch.
So, to beautifully summarize our micro to macro journey today, starch and glycogen both use alpha-glucose to create easily broken down energy storage. Cellulose uses beta-glucose to build rigid, straight chains that lock together for structural support. Just notice how a simple, tiny switch from an alpha-glucose to a beta-glucose monomer fundamentally shifts the entire purpose of a carbohydrate, from being an accessible energy battery into an impenetrable suit of armor.
Which leaves us with a pretty wild thought to end on. Because of that straight, tightly bound beta-1,4 linkage, humans completely lack the digestive enzymes required to hydrolyze cellulose. We easily break down the alpha bonds in starch, but cellulose just passes right through us as dietary fiber. But imagine if we could. What if human biology possessed the enzyme to break those beta-glycosidic bonds? We could literally eat and extract energy from grass, leaves, and the woody stems of plants. How would that reshape our entire relationship with nature and the global food supply? It's definitely an amazing thing to think about. Thanks for learning with me today, and I'll catch you on the next explainer.